Hello and welcome to Archaeology 101. Today's topic I have taken a childlike glee in creating, and that is the archaeology of poo. And what essentially I mean by that is looking at how archaeologists and scientists have been studying fossilized human and animal fecal remains known as coprolites or paleofeces and analyzing those to gain an insight into the human past. Coprolites and the more recently acquired terminology of paleofeces are just a word to describe preserved human or animal poo. Poo can be preserved all the way back millions of years ago, hundreds of millions of years ago, but there are also historic poos as well. They're an excellent repository for biomarkers when concerning things like health, diet, genetics. They can even detect things like environment as well from unconsciously intaken micro debris. But this all depends on the preservation of the poo. But there is so much to learn from old waste, as I will soon discuss. In our modern society, once we've gone to the toilet, we don't really think what happens to it very much. Our flushing toilets take our waste away into closed sewage systems, which are managed by surprisingly few people into our sanitized waste system. In the past, this was not the case. Most people would have left their leavings in middens or in cesspits, which are just holes in the ground or rubbish pits. Coprolites generally need a fairly rapid burial to be preserved, but this is not always the case. Caves are pretty good conduits to contain organic information because their environments don't tend to change very rapidly, so they're really good at preserving coprolites. So you can find coprolites in caves where someone has just chosen an area in the cave to do a poo, and that's been able to be preserved for thousands of years. Waterlogged environments are also really good, but usually only slow-moving waterways or stagnant waterways like bogs. These are really good for preserving coprolites as well, and I'll talk about these examples later. You might not have thought of this, but carbonization of coprolites is also a really good way to preserve coprolites, although it can damage the biomarkers and other organic remains within, but it does preserve them. And I'm going to talk about this really interesting case here. So if you see that middle image on the screen, that ceramic vessel there is an 18th century example from Lisbon, which was found from a floor within a house, which had burnt down during the 1700s. And within that jar, amongst other waste, was human feces. So this jar would have eventually, had the house not burned down, been emptied into a midden, cesspit, or perhaps into an open sewage channel. But it hasn't been able to reach that destination, and we have this really quite interesting insight into how people managed their waste back in the past, and it's been preserved for hundreds of years. I'm not sure how impressed the person who left that would have been, but I'm sure they had bigger problems at the time than what happened to their poo jar. Okay, so you've identified a coprolite in the field, you've successfully extracted it, and now you've got it back in the lab. What are you going to do with it next? You can do a surprising lot of different things to coprolites to get more information out of them. So one thing you can do, which is non-destructive, is scan it. You can scan poo. And you can gain a 3D image of it, but you can also find out the internal contents of that coprolite. So you don't have to destroy it, but you can see if there's anything worth extracting from that coprolite. There isn't always matter, organic residue, things like seeds or bones present within coprolites. They just haven't been preserved, or perhaps there just weren't any at the time. But now you've identified that there are without cracking that poo open. So you can now go on to do further analysis. Thin sectioning is quite common. That's taking a like it says in the name, a thin section of soil, say through a midden. You take a real thin section and you put that into a resin and you put it in on a glass slide, and then you can see the microstratigraphy of that midden layer, and within it you might be able to identify coprolites. And you can also see things like bone. You can tell from the shape sometimes what animal, or if it's a human coprolite, had deposited. Depositor is the name for the person who created that coprolite, and you can identify what depositor created that coprolite within that slide. You can also, from these slides, identify the presence of bone, seeds, pollen, etc. And you can determine things about what they've eaten, what the local environment might be, what they might be growing, and what they might be eating. Chemical and biomarker analysis has really come along since the late 90s, but has started to gain more traction in the 21st century. And what you're looking at is lipids, which come from both plants and animals, and these survive for millennia. They can stay around for a really long time because lipids don't tend to denature, and these can help identify, first of all, the species of the depositor, and then you can also determine what the depositor was eating from the presence of steroid signatures. Particular plants and animals have a certain signature. DNA has also been used as well to determine both who was the depositor and also what they might have been eating as well. We'll talk about the difficulties of DNA later. Back in the early 20th century, people might have done dry sieving, but this isn't tended to be used anymore, as dry sieving just destroys the coprolite and you don't get as much data as you might be able to through things like lipid analysis, so that's not really done anymore. So coprolite analysis is incredible, and I find it a little bit amusing that somebody's lasting legacy, they may not have made any physical impact on the world, but their number two has been preserved for someone like me to come along and analyze the hell out of it. The really impressive sample from the previous page, that coprolite in the top corner there, that is from Jorvik, from York, Viking York, and that is a whopping 7 inches long. That is certainly a legacy to leave. There's a funny story attached to that as well. When that poo was eventually deposited within the Jorvik Centre, it used to be passed around to visitors, which is hilarious. But then during the late 90s or the early 2000s, some unfortunate visitor dropped that coprolite and it broke. And now we can't go in and handle that poo anymore, so thanks a lot. We've all heard of that saying, you are what you eat, but I think it rings more true for you are what you poo. DNA can be preserved within coprolites, and the DNA can not only determine the species of the depositor, but also what that depositor had eaten recently. There have been some papers to suggest that we can determine sex of the depositor from DNA, but this seems to have mostly been achieved in non-human remains and doesn't seem to have been widely investigated more recently for humans. DNA is not stable, so that might be why, and it can break down easily in unfavorable conditions, even within the coprolite. And it's especially poor for prehistoric samples. As time goes along, that DNA is more likely to break down. However, there are other possible routes to determine sex of a depositor using things such as hormone levels of testosterone and estrogen, which are left behind in the coprolite. This all depends on the preservation of the coprolite, and these things do tend to break down, with the exception of lipids, which are the more hardier biomarkers, which you can see down there on that image by Shalito et al. From 2020. They're using sterols and bile acids to determine the species of the depositor, but you might not be able to use such as testosterone to determine sex, so that might be something that's investigated more in future, which I hope it does. DNA can also be used indirectly to look at the gut bacteria, the biome of the depositor as well. And a study recently was used to show that biomes of people in the past, as you might expect, was very different across the world. And it's very different culturally today as well, because diets vary and that promotes or inhibits certain bacteria. And we're beginning to understand more and more that these gut biomes, they have quite a big impact on your overall health and well-being. So this is the future of understanding people's well-being in the past. Coprolites are the greatest indicator for diet. They're better than indirect lipid analysis on things like ceramics. Whatever that person's ingested, whether intentionally or unintentionally, it's going to come out in the coprolite, and that can tell you about their diet, but also what's going on in the ecosystem around you. All the time you're breathing in and unintentionally consuming pollen grains, and even though you are perhaps intentionally eating a salad, taking in those pollen grains, you are taking in free flowing coprolites as well, which are just within our breathing environment. So you can identify things like grass grains, for example, grass pollen grains, which show you, oh, you're in a grassland environment, or there might be tree pollen in there as well, which suggests what sort of wooded areas are around at the time that person deposited. Lipids, as we've shown before, are really good at indicating diet. How these are preserved is during the fossilization process, they're converted into a more stable variety than when they were consumed. And they're so stable that they can survive for millions of years in the right circumstances. So that image on the bottom left-hand corner is a Carboniferous coprolite. That's hundreds of millions of years old, and Tripp et al. In 2022 were able to extract lipids from that coprolite, and that's really exciting. We're able to look at diets millions of years ago. And you can even distinguish plant and animal lipids from each other, and you can even look at what species of plant and animal that lipid came from as well. You can look at things like carbohydrates and proteins as well, but these are much less stable than lipids, and they're not as well preserved. So technology will allow for that in the future, to get carbohydrate and protein analysis down, but mostly people focus on lipids and sterols. You can get macrofossils within the coprolite as well, so diagnostic skeletal evidence can be extracted of bones and show what little animals that people have been eating. I'm going to point you to the bottom right hand corner image there. That is a snake fang. That is a western diamondback rattlesnake fang, and you can see the poison channel right there on the end of the fang, and that's from Lower Pecos Cave in Texas. Somebody, some mad lad, consumed nearly an entire rattlesnake, teeth and all, and then later pooed it all out. And I hope that was a painless process. Just look at that fang, that is sharp. But that brings into question, why would somebody want to eat a rattlesnake? It's very dangerous, but it also suggests they probably weren't doing this for caloric need. You wouldn't eat an entire rattlesnake with its fangs. This suggests that there's something else going on. This was probably part of some ritual or religious, spiritual process rather than because they were hungry. And you can find very often that snakes in Native American mythology are quite important. They're ill omen, so maybe this was something to do with that. We must keep in mind when looking at coprolites that just because the bones, the pollen, the lipids are within that coprolite, those weren't necessarily consumed on that day. If this video wasn't already gross enough, let's move on to possibly my favorite part about coprolite analysis, and that's parasites. Parasites are an ongoing problem. We will probably always have parasites living with us, and we still have them in the modern day, even though we have been able to reduce parasitism through things like closing sewers and more hygienic practices, washing hands, not eating raw or undercooked foods, etc., etc. But parasites were a very common issue in the past, and indeed wherever you seem to find coprolites, you will find parasite eggs within them that show that people are being plagued by things such as tapeworms, roundworms, whipworms, and flukes. And let's talk about a very famous example here, and that's Must Farm, which was a late Bronze Age village which burned down in East England way back in the Bronze Age. And they preserved all sorts of organic remains, but due to the slow flowing water, we were able to extract human and dog coprolites from where people had thrown their feces into the water. And the example that's highlighted in red on the diagram there is an unfortunate individual who has 4 different types of parasite eggs in their coprolite. So this person was absolutely riddled with parasites and probably was not feeling too well at the time. Also, the bottom example is a dog coprolite, and that dog had 5 different types of worm eggs within its poo, and that's a very sick dog. So we can see from the environment that's preserved the coprolites really well, and shown that people have been absolutely plagued by mostly aquatic occurring worms due to them probably eating things like frogs or fish. However, the upside is that living by the water may have saved these people who lived in this village from ground-dwelling worms, such as types of roundworms, which don't occur in water. So they may have been saved one type of parasite for another. Worms have also been found within coprolites preserved in the late Neolithic henge of Durrington Walls in Wiltshire. However, interestingly, the parasites found within the coprolites here come from cow flesh and aren't usually found within humans, so this might actually be a case of false parasitism. These types of parasites aren't designed to live within human hosts, so they may have just consumed the eggs unwittingly and then passed them without any ongoing issues. They may have had one bout of illness and then it was over, or they just might not have realized at all and they just passed the eggs out without any problems. Not only are worm eggs found, but now we are being able to extract bacteria and viruses which are preserved within the coprolites as well. So an example from 600 to 800 AD in La Cueva de los Chiquitos, this cave in Mexico, C. parvum was extracted from coprolites, and C. parvum is a diarrhea-causing bacteria, and this seems to have passed through many of the individuals, or potentially one individual, within this cave. And C. parvum can pass very easily between hosts, especially in unhygienic conditions where you don't have hand washing or closed sewage systems. Parasites are actually making a bit of a comeback in the modern period. Parasites which weren't so much of a problem are returning. Certain types of salmon, smoked salmon, can contain worm eggs which can then infect you because these salmon are not cooked in the same way and those don't destroy the parasite. So think about that the next time you're going to consume a piece of salmon. With any scientific analysis, you're going to get innate problems during the analysis. So feces can sometimes be confused amongst depositors. Dogs, pigs, and humans especially can sometimes be difficult to distinguish because they can eat very similar diets. This is less of a problem now with steroid studies because you can identify the exact species. However, dogs can muck this up. Dogs are gross and they eat poo. They're coprophages, and this is especially confusing if they're eating human feces. It makes it seem like dogs are actually eating people themselves. This might actually be occurring in very rare instances, but most likely if you're finding a dog which has a profile that makes it look like it's eating humans, it's probably eating human coprolites. Diet, as I discussed briefly earlier, is also seasonal, and it doesn't always present the entire dietary picture of a human's lifetime. It just shows you that snapshot of what that person has been eating over the past few days. We've also discussed earlier micro debris. Pollen is especially a big offender here because you unintentionally consume pollen, but you can also intentionally consume pollen during eating greens, vegetables, etc. This confuses dietary reconstruction, and it can take up to 30 days to pass pollen from your system. So long scale pollen analysis from coprolites can be tricky. Samples can also be contaminated as well. They're not only affected by underground processes such as taphonomy, diagenetic processes which destroy or contaminate certain parts of the coprolite. During laboratory analysis, if you don't take the right precautions, if you don't wear gloves, if you don't wear a mask, you can put in modern data into that coprolite and you might not understand, and you might confuse the picture, and we won't understand completely the past of that coprolite. Coprolites also don't always contain the relevant information either. During the preservation process, they might not have the particular biomarkers available, or simply they might not have consumed the right product which will preserve, depending on dietary intake. So let's wrap up on what we've learned about poo. So coprolites or paleofeces are a fulcrum of biological information. They give us really good insights into the health, diet, environment of depositors in the past, and it can also help us identify the species of that depositor as well. The problem with coprolites is that they've not historically been recognized in the field physically. An archaeologist may not understand what they're seeing if they're not an expert in coprolites and throw it away. Indeed, when I was in the field, if I ever had seen a coprolite, it ended up on a spoil heap, I would not have recognized it. It's a very underutilized area historically, but there are groups of archaeologists with a good sense of humor here. The Coprolite Research, Archaeological and Paleo-Environmental Potentials Group, aka CRAP, are at the forefront of coprolite research and are trying to get more and more archaeologists to recognize coprolites and get more analysis done on them. We have a very good sense of humor in archaeology, especially apparently to the people who look at prehistoric poo. However, where we can actually get coprolites and when we get them into the lab, they're not always optimal to extract biomarkers or DNA simply because they do not preserve. So even though they can be really good at getting all this information, it's not always viable. Thank you very much for watching me ramble on about poo. I hope you enjoyed yourself as much as I did researching this video. I learned a lot, I had a giggle, it was a good time. I'm going to leave the sources and the figures in the description as I always do, so you can go ahead and look at those sources for yourself to learn more about historic poo. I would recommend starting with Shalaito et al.'s 2022 paper because that gives a really quite easy to read description on both the history of coprolite research and where it's going in the future as well. So I will put that at the very top for you to read. Thank you very much, and I'll see you next time on Archaeology 101.